Hello everyone, today we are going to master about factorial completely randomized design, also known as Factorial CRD. Before starting this topic, I would like to ask this. In a real agricultural field, does a plant ever just receive one variable at a time? Of course not. Plant receive a changing level of temperature, moisture, humidity, and nutrient level, and so on. In this image, in the left hand side, a plant is receiving only one variable at a time. But in the right hand side, plant is receiving a varying level of pathogen, water, light, and so on. This is where we need the concept of factorial. This is factorial reality where the plant receive a multiple factor simultaneously at a time, and we can evaluate the multiple factor together. How should we proceed about the randomization of our treatment? Today we are focusing just about the completely randomized design. In a completely randomized structure, if our experimental unit is relatively homogeneous, or there is no any noticeable change in gradient or soil type or slope, by completely randomizing our treatment throughout our experimental unit, we ensure that every single pot or plot has an equal mathematical chance of receiving any treatment. I can simply just illustrate this through the layout of the experiment, and I can give a quick look of the CRD and RCBD layout. So this is the CRD design where we randomize all the treatment. Here is the in this experimental experiment there is 4 treatment, T1, T2, T3, and T4 with a 5 replication, and we randomize all of them throughout the experimental unit. We can see T1, 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 T1 repeated 5 times, and it is randomly assigned. But in case of RCPD, when there is heterogeneous field condition, there is a gradient or so different soil type and so on, we capture the variation of the field with the blocking factor. We add the blocking factor, and block acts as a replication. And within each block, there should be every treatment we have. We had 4 treatment, T1, T2, T3, T4. So every block should have every treatment. Here also we can see T1, T2, T3, T4, and we should randomize the treatment within the block.
In this study, instead of looking at one factor at a time, we cross two main factors to create a different treatment combination. Our experiment include factor A, which is the rice cultivar, includes CL18 diamine ozark, factor B, the nitrogen rates, arranged from 0, 50 to 100 kilogram a hectare, and our response is the grain yield. So the main goal is to evaluate both the individual effect of cultivar and nitrogen, and also determine if there is an interaction between them. So here we structure the experiment by combining all the level of both factors. So we have 3 cultivars, CL18 diamine and ozark, times 3 nitrogens rates. So that's equal to 9 treatment combination. This is what we call factorial design. The design is powerful because it allows us to test not only the main effect, but also whether the response to the nitrogen depends on the cultivars. So one major advantage of this design is statistical efficiency. So we get more information from the same experimental comparing to do studying each factor separately. So it's allow us to test for the interaction, which is help answer our practical question like, does the nitrogen rate affect each cultivar differently? And also it has a hidden replication, more powerful, and reduce the number of experimental units. However, there are some limitations as we add more factors or levels, so the number of the treatment combination increase quickly, which is make the experimental more resource intensive. Also, without enough replication, it may reduce our ability to detect significant effect. So, to analyze the data, we use this model. So, Y is the observed yield equal to U is which is the overall mean, plus alpha, which is factor A, the effective cultivar, plus beta, which is the factor B, the effective nitrogen, plus the interaction between them, plus the random error.
And I'll share with you the results of our exploratory analysis of the effect of cultivar and nitrogen level on rice yield using a factorial CRD. Just to remember, we have 3 cultivars, 3 levels of nitrogen, and 4 replications. After loading the packages and reading the data, we can see that the experiment is balanced because we have the same number of observations for each treatment combination. And from the descriptive analysis, we can see that the yield increases with the nitrogen level for all the cultivars. For example, if we take the first cultivar here, we can see that this cultivar had the highest yield when we used the highest level of nitrogen of 100. This histogram here shows us the distribution of the yield data. Here we have the yield data on the x-axis, and here we have the frequency on the y-axis. And we can see that the data is well distributed with no very strong outliers. This box plot here compares the yield by cultivar, and we can see differences in yield among those cultivars, indicating a potential effect of cultivar on yield. In this case, we can see that the cultivar diamond, represented by the orange bar here, had on average the highest yield, while the cultivar ozark, represented by the purple bar, had on average the lowest yield. The second box plot here compares yield by nitrogen level, and we can also see differences in yield among those different levels of nitrogen, indicating a potential effect of nitrogen on yield. And we also observe that the highest yield occurs when we use the highest level of nitrogen of 100. This third box plot here compares yield among those different treatment combinations. Since we have 3 cultivars in 3 levels of nitrogen, we have a total of 9 treatment combinations. And we can see that some treatment combinations produce higher yield as compared to other treatment combinations. In this case, we were able to see that the combination of the cultivar diamond with the level of nitrogen of 100 produced the highest yield on average. With this interaction plot here, we can see that the lines are not parallel, indicating a potential interaction between the factors cultivar and nitrogen level, meaning that the effect of one factor is not the same across all the levels of the other factor. For example, here we can see that the yield of the cultivar diamond, represented by the orange color, increases more with the nitrogen levels compared to the yield of the cultivar ozark, represented by the purple color.
The first step in analyzing the data is to treat all of the necessary elements as a factor. In our case, I have treated the cultivars and the nitrogen rates as a factor. After that, I check to see if the data is balanced or not by running this code. And it was indeed balanced because each of the treatment combinations appeared 4 times in our data. Then as for the model, I use the AOV function and put the model as yield as a function of cultivar, nitrogen, and also their interaction. And as you can see on the screen, it was highly significant under the p-value of 0.001, but we cannot be certain that it is indeed the correct result before we check the assumptions. For that, I use both the plots and also the formal tests. Looking at the plots, first of all, we can see that the residuals are homogeneously distributed, but the QQ plot shows that the residuals are not normal. But I did not run the Box-Cox transformation because ANOVA is somewhat resistant to non-normal residuals. If the residuals were heterogeneous, we would definitely have to run a Box-Cox, but I checked that using the Shapiro and the Levine test. The Levine test was not significant, meaning it was indeed the homogeneous, the residuals, and the Shapiro test was significant, meaning that the residuals were not normal. Moving on to the post-hoc test, I used the Tukey's HHT test, and it gave me a CLD table. And since our interaction was significant, we cannot interpret each of the factors separately. We have to do them in a combination. So here, under the nitrogen 0 kz per hectare, we can see that Ozark was the worst performing with the lowest E means, but CLL 18 was the best performing. But then under the 50 kilograms per hectare nitrogen dose, Diamond took the lead with 5,275. Ozark still remained in the last position, and then CLL 18 was the second place. Finally, under the 100 kilograms per hectare nitrogen rate, Diamond, in this instance too, took the lead at 6,182 kilograms per hectare, and Ozark was the least or the poorest performing among the three genotypes.